Welcome everyone to our 30th Bioelectrodynamics webinar series. I would like to give some small housekeeping notes. As you'll see by the permissions of this webinar, during the talk, all the attendees are muted and have the video off. If you have any questions, feel free to type them to the chat or to the Q&A opportunity here. There's application for it in Teams, and the talk will be roughly 45 minutes. And we have all these recorded and will be later on, including the Q&A, published on our YouTube channel. I would like to, before I introduce the speaker, I would like to emphasize that this webinar is supported by the International Union of Radio Science, URSI, as well as Engineering in Medicine Biology chapter of the Czechoslovakia section of IEEE. And I would like to also highlight that particularly this week is special because it's the so-called Biophysics Week organized by the Biophysical Society. So I'd also like to take the opportunity to emphasize this. Today's speaker, I'm very excited to introduce him, is Dr. Rui Rodrigues. He earned his master's in biological engineering in 2011 with his dissertation in Karlsruhe Institute of Technology in Germany. And from 2012 to 15, he worked as a research fellow on two R&D projects, mostly focusing on development and implementation of novel technologies and products for the food industry. From 2015 to 2019, Dr. Rodrigues developed his PhD project at the University of Minho in Portugal and Aalborg University in Denmark, focusing on the study of electric fields on protein structure and their function. And since 2020, he is had a position as a postdoc and a researcher at the Center of Biological Engineering. And since 2023, he's assistant researcher at the same institution. So without further ado, Rui, the floor is yours.
So, hello, good morning. Thank you so much for the opportunity of being here. I'm really excited about this talk. I'm trying to share my presentation. I don't know if it's on yet.
We're waiting for it. It looks like it's going. Good. Yes, yeah. Please go ahead. Just build the full screen. I think we'll be ready.
Perfect. There we go. So thank you so much for the invitation, and thank you for the opportunity of sharing my research. And I also would like to acknowledge all the assistants. So today I'm talking about my research about moderated electric fields in proteins. This is the outline of my presentation. I will start with a brief introduction, some background and some explanation what is moderated electric fields processing. Then I will address the effects of math in protein unfolding and conformation, also protein interactions, protein-protein interactions, aggregation and gelation, and we'll end up with some conclusions and future perspectives. But first, I come from the University of Minho, which is located in Braga, a city in the northern region of Portugal. Braga is a historical city with over 2,000 years of history, a lot of historical sites and nice buildings, but we are also located near the Portuguese most important natural park, so natural wonders are around the corner as well. University of Minho is a young university, just turned 50 years, but it's quite dynamic and it's recognized internationally by its quality, especially in research. I conduct my research in the Center of Biological Engineering, and about this center, we're about 300 members, including faculty professors, researchers, students, among PhD and master students, external collaborators, and technical staff. And our research areas are divided in four main focus: food biotechnology, industrial biotechnology, environmental biotechnology, and health biotechnology and bioengineering. We are also a quite new and dynamic center, and just last year, we reached the mark of 10,000 publications. Now, going to the topic, I'm going to talk to you about moderated electric fields, which is sometimes known as ohmic heating. And how to explain this process? Well, we can consider a simple electrical circuit where through a power source we apply a voltage, V, that goes through the circuit, and involves the passage of a current I. And if you place an electrical resistance, we are able to dissipate energy in the form of heat in this resistance. So this is a very simple diagram of the process we are dealing with. Taking this analogy, in moderated electric field, we usually use AC power supply. And instead of the resistance, we use a conductive sample. This conductive sample can be any foodstuff, any biomaterial, can be solid, liquid, a mixture of both. As long as it has some water and electrolytes, current will flow through and it will react as a resistance. We apply this current by means of two electrodes contacting these samples. These electrodes are usually made of stable materials such as stainless steel, platinum, titanium, and so on. And by this means, we are able to process the sample, either food or a biomaterial. And the process can be described in a very simple and straightforward way. The energy dissipated from electrical energy to thermal energy, it can be defined by the voltage, the resistance, and the current flowing through the system. Of course, electrical conductivity is a very important parameter, and it also can be defined by the resistance of the material, the section area, and the length of the sample. And we also know that the electrical conductivity changes within the temperature, usually increases linearly with the temperature, and we can also define that on a quite simple way. But why are we
Rui, you got muted.
I'm sorry.
Yeah, okay, you're back.
Where did you lost me?
Just sliding, beginning of this slide.
Okay.
Why are we?
Okay, so I'm sorry for that. So the definition of moderate electric field technology, we usually involve electrical fields ranging roughly from 1 volt to 1000 volts per centimeter. Because of this electrical current, electrical energy is dissipated into heat, usually isotropically, meaning it's mostly homogeneous. There are some exceptions, but usually you can consider that there's a fast and homogeneous heat dissipation. And of course, this offers some processing advantages in terms of energetic and heating efficiency. For example, we can easily achieve several volts, if not a few tens of volts, several degrees, or if not several tens of degrees of thermal increase per second. And in terms of energetic efficiency, we can reach conversion efficiencies well above 95%. Just to put in perspective, a thermal process that comes from a combustion usually does not surpass 50 or 60% of energetic efficiency. And of course, we have the presence of an electric field, and electric fields interacting with the biomaterials and biological molecules, naturally, they bring implications. So we know, I'm so sorry, I'm having some troubles with the share. Here we go again. Sorry about that. So when we use a protein solution as our resistor, and we apply an electrical field with associated voltage and frequency, we are expecting to observe some effects. And why are we interested in probing the electrical field effects in protein? Because it can affect protein functionality. Of course, we know that proteins have intrinsic functionalities, such as catalytic, as is the case of enzymes. They can bind and transport some bioactive compounds and drugs, and they also play a role in immunology, for example. But apart from these intrinsic functionalities, we can actually induce protein functionality. And how we do that? Usually, we change the native fold of the protein to expose reactive groups, such hydrophobic groups or cysteines. And to induce these new functionalities, we have to disrupt the native structure, either by a chemical agent or a physical stress, such as heat or an external electrical field. And by unfolding or changing the native structure of the proteins, we can promote new interactions. And this is the precursor, of course, for new bioactivities, to formation of protein complexes, protein gels, protein emulsions, and so on. So our main focus will be to study the electrical field effects and how they can control protein functionality. But when we consider electrical field effects in proteins, we can have two approaches. We can consider a DC circuit where the electrons flow on a single direction. And of course, considering DC current, we have to consider electrical polarization. When we have dipoles at rest, they are not particularly aligned, but when we apply an electrical field, the charges align with the field, changing not only the dipolar moment, but also can imply changes in molecular conformation, displacement, and so on. And as we know, proteins can be considered as a dipoles because they have a charge distribution across their structure. And when we apply an electric field, the protein as a dipole will align with that field. The consequences are not just the alignment of the protein, but of course, some forces are applied into the protein structure. With the direction of the field, we have the electrostatic force, and the opposite direction, we have frictional force and electrophoretic force. And of course, all of these will have consequences in protein structure and also in its dynamics and functionality. And this has been actually the subject of several studies, applying strong electric fields or strong DC electrical fields, as is the case of pulsed electric field processing, or in many studies of molecular dynamic simulations, where the application of electrical fields are simulated in the protein structure. And in these kinds of applications, sometimes tens of thousands or even millions of volts are used to produce these results. What researchers have is that this strong polarization will imply the unfolding of the protein, and of course, following consequences as protein aggregation and loss of solubility, and so on. And in this case, because we have a unidirectional electrical field, the effects observed are proportional to the intensity of this field. So if we increase the voltage, we'll increase polarization, we'll increase the electrostatic force, and we'll increase molecular displacement. Another scenario is when we use AC electric fields, because the alternating current will imply that the electrons or the charges are not flowing in one direction, but either oscillating. And when we apply an oscillating electric field into a protein, for example, when we apply a low frequency electric field, the protein will align towards the direction of the field will invert, and the protein will try to align on the other direction. By using a low frequency, we give time to the protein molecules to rotate and fully adjust to the direction of the field. But considering electric fields of high frequency, the polarity changes will be much faster, and the protein will not have time to fully align. So instead of half rotation, we will induce molecular vibration. And there's a particular case when the frequency will be similar to the amplitude of motion. And in this particular case, the protein will align, and as the field inversion will coincide with the maximum, the half rotation, the protein will keep rotating and not reversing its direction towards the electrical field. These data of protein mobility and protein vibration were actually obtained by Professor Sastry and his group. Professor Sastry is a pioneer of electrical field processing, and they actually were able, through means of simulation, to determine the displacement of proteins by subjected to alternating current electrical fields. And here we have an example of his research, where he proves that low frequencies provoke high displacement, and high frequencies result in lower displacement, meaning molecular vibration instead of half or full rotation. Another group on an independent research by Beckard and Dustin, addressing a similar topic, actually were able to calculate the frictional force involved in AC fields and proteins. And they simulated the frictional force involved by applying just 5 volts per centimeter, which is a small electrical field, for 1 second into lysozyme and BSA. And they determined that the energy dissipated by this molecular motion was 850 kilojoule per mole for lysozyme and 45 kilojoule per mole for BSA. Just as a standard or comparison, I included here the energy involved, for example, in hydrogen bonding in alpha helix or beta sheet that range from 8.1 kilojoule per mole or 6.6 kilojoule per mole. So it's clear that even applying a small electrical field for a small period of time, we are dissipating enough energy at least to break hydrogen bonding and possibly other intermolecular bonds within the protein structure. So against this background, and since we are working with electrical field processing for food and biotechnological applications, we actually came up with a lot of questions, as the one stated here. So we wanted to know what is affected by the moderated electric field treatments, how protein structure can change, how we can change protein interactions, and for example, influence processes such as protein aggregation. We also wanted to know what's the role of moderated electric field variables. For example, what happens if we change the electrical field strength or the electrical frequency, and how these changes can impact protein-based systems, meaning how can we change physicochemical and functional properties of proteins and protein-based systems. So we started by probing how moderated electric fields interfere with protein denaturation. Since this technology is mostly applied as a thermal technology, protein denaturation is a big topic. To conduct this study, we used beta-lactoglobulin. It's a small globular protein found in milk, and it's actually used as a model protein because it's very well studied. Its structure is well characterized. Its thermal stability, structural dynamics is very well described. So it's the perfect candidate to conduct these studies. And how can we study the protein denaturation process? Well, actually, we choose two main techniques: fluorescence spectroscopy, where we probe the fluorescence of aromatic amino acids within the protein structure, and we know that as the protein unfolds, the fluorescence of these amino acids will shift, either increasing its intensity and shifting on the maximum peak due exposure to solvent, change of microenvironment. And also we used circle dichroism spectroscopy, which is a technique very sensitive to protein secondary structure. And by following, for example, the changes on the spectra with temperature increase, we are able to trace an unfolding or a denaturation profile. And to do so, we actually chose to perform in situ determinations. The way to do that is actually to construct a simple setup where we adapted a spectroscopic cuvette. We fitted 2 platinum electrodes and we connected that to a power supply. We actually use a common function generator that it produces low voltage up to 10 volts, and it's an output of 50 ohm or a maximum of 1000 milliamperes. So this is actually a really low intensity, low power electrical field. In fact, this setup was not able to produce thermal increasing within the cuvette. So we actually used the Peltier elements on the spectroscopic equipment to control the temperature. And we decided to run some temperature screens alongside with applying some electrical fields. And for example, following the fluorescence spectroscopy of the protein, we used just a thermal action or applying an electrical field of 20 kilohertz and 10 volts, and we can actually follow the unfolding profile of the protein. And you can see here above 40 Celsius, where the protein conformation starts to change, we see small differences between the path of the conventional heating process and the electrical field induced process. We also follow the cooling down or the refolding of the protein, and here we didn't see much differences between the processes. We did the same using circular dichroism spectroscopy, and here right away, from the moment we start applying the electrical field, we start to see some differences on the transition profile between the process that had the electrical field and the conventional heating process, which not included the presence of the electrical field. Following this, we decided to explore this kind of approach. And on the next few slides, I'm going to focus on circle dichroism because it seemed that it presented the better results. I'm sorry. And also, it's a more sensitive technique to structural features of the protein. So next step, it was to induce these thermal transitions alongside with the presence of the different electric fields. In this case, we changed the electrical frequency. And we can see here the line in black. It's a conventional thermal process, no electrical field involved. And the different colors, we applied an electric field of 10 volts per centimeter at different frequencies. And we can see here the lowest frequency tested, 50 hertz. We have a big change on the transition profile. And as we increase the electrical frequency, the profile gets closer and closer to the control without the electrical field. But even the higher frequencies applied implies some changes on the transition profile. And we are able to calculate the midpoint transition of this profile and actually calculate the melting temperature of the protein subjected to different electrical frequencies. And you can see that here on this insert. This line represents the melting temperature of the protein, which is about 74 Celsius. And we here represented the melting temperature of the protein when exposed to 10 volts per centimeter at different frequencies. And you can see that for 50 hertz, we decrease the melting temperature about 20 Celsius. And as we increase the frequency, the melting temperature gets closer and closer to the conventional one. And actually, when you increase the frequency to the kilohertz or megahertz range, we were actually able to delay that melting temperature by a few degrees. Using a similar approach, we tested the influence of different electrical field strengths. So we took our electric field at 50 hertz, and we changed the voltage applied. And what we observed is with the increasing of the voltage, there's an increasing on the actually decreasing on the melting temperature, and this is a linear relationship. So it tells us that increasing the electrical field will linearly increase the effects on the protein structure, contrary to the frequency effect, which is not linear. We also took a look at the protein structure after we conducted these processes, and you can see, comparing to the native protein, all the processed samples lost secondary structural elements, namely alpha helix and beta strand. Our control, which was just the thermal process, we lose about 2% of helical content and 20% of beta strand. Of course, those being transformed into thirds and random coil. But both processes where we applied an electrical field, the structural loss was higher, and this is particularly evident when you use low frequency. We lost about 7% of alpha helix and about 30% of beta strand, of course, increasing random coil and turns. And these consequences are not just on protein structure or secondary structural elements. We also probed another structural features. For example, we use ANS as a conformational probe. ANS usually binds to hydrophobic sites on the proteins. So we observed that thermal processing increases the surface hydrophobicity of the protein, but this is particularly relevant when you apply an electrical field. And you can see here both electrical treatments increase the surface hydrophobicity of the protein with particular focus to the low frequency one. This also implies an increase of the binding properties because more hydrophobic sites are available, so the protein is able to bind more hydrophobic molecules. This also changes the sulfhydryl reactivity because these conformational changes also impose exposure or occlusion of free cysteines, and same thing with the tryptophan residues. Following this, we have established that the presence of electrical fields and different electrical frequencies can change protein structural features, and we decided to probe that in protein interactions. And we know that beta-lactoglobulin is able to bind small hydrophobic compounds on its barrel structure. And we decided to probe that with the phenolic compounds. And
Rui, got muted again?
Yeah. Again, okay. Sorry.
No problem.
When we apply this kind of processing, just a thermal process with an electrical field, and we probe the affinity of the beta-lactoglobulin to some phenolic compounds, we observed that a partial denaturation increases the binding capacity because those hydrophobic sites became more available. But when we do so with a present electrical field, actually you can increase the binding affinity of this protein to some phenolic compounds. This proves that the structural changes are the influence of the electrical field during the structural transition actually results on a more open-like structure, more willing to interact with the hydrophobic molecules. On the same way, we decided to probe, for example, antioxidant capacity of these compounds when bound to the protein. And we observed that the processed protein just by conventional method, meaning just thermal processing, did not display any difference on its antioxidant capacity regarding the control, which is the native protein, but both proteins processed under the electrical field displayed higher antioxidant capacity. Of course, then you can conclude that protein functionalization through electrical field can increase the affinity for phenolic compounds. And actually, we have been observing that for other classes of hydrophobic compounds as well. So if increases the affinity, higher affinity also implies higher antioxidant capacity among other bioactivities. And once again, we observe that treatments at low frequency are more effective in these modifications. Another topic of interest, and we know that when we are dealing with protein structure and protein interactions, immunoreactivity is also a big topic. And since we are working a lot in food technology, the immunoreactivity and allergenicity of some food proteins are of high interest. And we are applying electrical field processing in some common food proteins. Beta-globulin is one of the examples, but we also use soy proteins, which are quite known food allergens. And what we have been learning is that the structural changes imposed by the electrical fields can change the denaturation pathways of the protein, can also interfere with the aggregation pathways. And these, of course, will have an impact on how these proteins are digested, how they will reach our intestine, and how they will interact with the antibodies and dissolve allergenic responses. Allergenicity is quite a complex biochemical process, and so far we have been focused just in the immunoreactivity, meaning the recognition of these proteins by antibodies. And despite we don't have a really straightforward or conclusive results, for example, we are not able to link increasing electrical field strength with the reduced reactivity, but we are able to find specific conditions where applying electrical field treatments at certain frequencies, at certain temperatures, we were actually able to decrease the immunoreactivity of these proteins by much. So this is just the first step. It's we have a couple of papers published on that, but it's still preliminary. But we are trying to explore how these structural modifications induced by the electrical fields can actually modulate the immunoreactivity of these proteins and possibly mitigating or eliminating allergenic responses. Another interesting topic is enzymatic activity on the presence of the electrical fields. Here I bring an example again from Professor Sastry. Actually, this is the same study where they modulated the molecular displacement of these proteins under electrical fields. And what we can see here is studying the activity of alpha amylase on a broad range of frequencies from 1 Hz to 1 megahertz at a voltage of just 1 volt per centimeter, comparing to the control where no electrical field was applied. Using low electrical frequencies, the enzymatic activity increased up to actually 40%, but keeping increasing the electrical frequency actually resulted on the small reduction on the enzymatic activity. On a following study from the same group, they decided to probe the activity of another enzyme, in this case cellulase. They applied electrical frequency of 60 Hz, and they changed the electrical field intensity. cellulose has an optimum temperature of about 50 Celsius. And what we can see here by the results is suboptimal temperatures, the presence of the electrical fields at different intensities actually results on enzyme activation. This is probably because the induced molecular motion actually increases enzymatic activity by increasing mobility from the protein structure and mobility of the substrate species. When we reach the optimum temperature of the protein, the lower electrical field didn't seem to display a lot of effect, but the higher electrical field actually seemed to cause some enzyme inactivation. And when they tested above the optimal temperature, where thermal inactivations already started playing some role, they actually could not get any activity measurements from the higher electrical field, and the lower electrical field actually decreased the enzymatic activity a little bit more. So these again are consistent results with previous observations, where the molecular motion and additional energetic dissipation imposed by alternating electrical fields actually play a role, in this case, in enzymatic activity. Of course, in our group, we also decided to study enzymatic activity. In this case, we are looking into pectinase activity, and we applied an electrical field of 7 volts per centimeter, again, a really low electrical field, and we started using 20 kilohertz. And we had similar observations where at suboptimum temperatures, the presence of the electrical field seemed to increase enzymatic activity. And as we approached to the optimum temperature of the enzyme, we no longer observed differences between our control or the enzyme exposed to moderate electric fields. We also decided to probe the effects of the electrical frequency. And what we observed is around optimum temperature, low electrical fields result in high enzymatic activity, but as we increase the electrical frequency, the enzymatic activity was reduced. And the opposite was true for suboptimal temperatures. When you apply these treatments at 20 Celsius, we see actually an increase in the enzymatic activity with increase of the electrical frequency. Another topic of our interest is protein interactions, protein aggregation, and network formation. These processes are actually pretty well described in the literature. So as a protein unfolds, some reactive groups are available, so proteins can interact and aggregate. And depending on the extent of these structural changes, and also some environmental conditions such as pH, presence of salts, or temperature, these aggregates can assume different morphologies as linear fine-stranded aggregates or more amorphous or globular aggregates. And if the protein concentration is high enough and we propagate this process, we can actually obtain gels. And the same thing stands for the gels. We can get highly ordered, fine-stranded structures or more globular or amorphous gels. And this, of course, will have impact on their physicochemical characteristics and also functionality. So we decided to probe the effects of MEF processing in protein aggregation. In this case, we used whey proteins. And when we took a protein solution and applied processing, in this case, it was 85 Celsius for some time, applying just a conventional treatment where just temperature was involved, and this MEF treatment where we use the same thermal profile, but with the presence of an electrical field. And we can see that the aggregation kinetics is changed. And as we increase the electrical field strength, we actually change the size of these aggregates. Using just a thermal process from the target temperature up to 5 minutes, we actually went from 1000 nanometers to more than 250 nanometers. But using electrical field, we are able actually to keep the size of these aggregates almost constant through the process. And these consequences were not only on the aggregation kinetics and also aggregate size, but also on these aggregates shape. Using electrical fields, we can see that we can change from spherical aggregates to something more elongated or fibrillar-like. This is another example of a higher protein concentration, almost forming a gel. And when we just use temperature, we get this compact, dense, and amorphous structure. using an electrical field, we have a more fine-stranded and more linear-like aggregates in these samples. We decided to keep further exploring this, and the topic of interest is protein fibrillation. And just a small introduction about the topic. Amyloid-like fibrils are known as a pathological aggregates in human neurodegenerative diseases, such as Alzheimer or Parkinson. But recent studies have been finding that fibrillation is a generic property of pretty much all proteins. And amyloids derived from proteins, for example, food proteins, are of a lot of interest for biomedicine, tissue engineering, environmental science, material science, or food science. These are highly ordered structures and highly stable. And how are they formed? Starting from a beta strand from a protein, we know that it can be associated to beta sheet, or higher levels of association can produce cross-beta structures. If these cross-beta structures further associate, they will form protofibrils, and these protofibrils will further associate and mature into amyloid fibrils. So this is a quite complex and highly ordered process. And we decided to probe protein fibrillation under the presence of an electrical field. And I would like to apologize in advance. I will not go into much detail into this slide because this is unpublished research. But what we did, it was to promote fibrillation of whey proteins at the concentration of 10 grams per liter at 90 Celsius under conventional heating and under moderate electric fields using 20 volts per centimeter and 20 kilohertz. And we are able to observe differences on the formation, on kinetic formation of these fibrils and also on their morphology. Using a conventional heating process, we actually obtain linear, straight, mature fibrils. And when you use moderate electric field processing, we can actually see that these fibrils are thinner and more disordered. They resemble more protofibrils than mature fibrils. But in fact, we obtain those in higher concentrations than in the conventional heating process. So this suggests that the influence of the electrical field, probably resultant from the molecular motion, promotes higher formation kinetics, but the different morphologies of these fibrils, as they probably do not mature into amyloid fibrils on the same way. But again, these are still preliminary results, but quite promising. Another thing that we have been studying is the formation of protein gels. And what's our interest in protein gels? Well, they can serve as structuring agents for food products, for example, or as a support for cellular growth, or as agents to encapsulate and release drugs and bioactive compounds. So we formed some protein gels, in this case whey protein gels, under conventional heating and under heating by an electric field of 20 kilohertz and 50 kilohertz. And here we use rheology to characterize these gels. Just a quick note, rheology is a standard technique to probe the viscoelastic behavior of materials. And right away, we can see that these gels behave differently. They have different structural features. When we took a look at the microscope and we watched these gels, the gel produced by thermal action presented a compact and heterogeneous structure, and the ones produced under electrical field presented a more ordered and fine-stranded structure. Of course, this is in line with our previous observations with the protein aggregates. And this is particularly interesting for using low-frequency electric fields, where we have a highly ordered and fine-stranded structure. And it's not just structural features, but these gels presented higher elasticity. We are actually able to probe the interactions and the bonds involved on the formation of these gels, and the presence of the electrical field implied that less chemical bonds, for example, disulfide bridges, were involved, but more hydrophobic and electrostatic interactions were involved on the formation of these gels. They also presented other characteristics such as higher water retention capacity. And these effects were actually potentiated by the increase of the electrical field strength and were more evident at lower electrical frequencies. Another application of this gel structuring strategy was, for example, to the production of aerogels for scaffolds to be used in tissue engineering. How we managed to do that? We promote the formation of BSA and casein gels. We remove the solvent by freeze drying, and we created a porous solid-like structure with a high surface area. So here we have picture of the gel, and after we remove the solvent, we obtain our aerogel. And now we can see here some imaging of the surface of this solid. And again, we use a similar strategy, conventional heating as a control, and two alternatives of moderate electrical field processing at 20 volts per centimeter and another one at 40 volts per centimeter. And we can see as we use electrical field processing and increase the electrical field strength, the morphology of this material changes. Particularly in this case, where we can see a highly ordered structure with well-defined pores, and we actually can see the pores are well connected. So we also have a great interconnectivity. The materials produced under the influence of meth also presented lower hardness, higher elasticity, higher porosity, and higher swelling capacity. And as I mentioned, these materials were used as scaffolds for tissue engineering. So we seeded them with human fibroblast cells. And after a few days, we actually determined the number and the activity of those cells after 5 and 8 days. And again, a similar pattern. In all cases, the cells grow pretty well, but the materials produced under the influence of the electrical field, the number of cells and their metabolic activity was higher. And again, we observed a positive correlation with the electrical field intensity used during the production of these materials. And this brings to the end the presentation I had for you today. So our main focus is electrical field processing. Of course, we know that we can generate heat and reduce thermal load applying this kind of technology, but we also learned that we can promote molecular changes, modulate protein activity, and also impose changes, for example, in macrostructural level in protein-based systems. So the take-home message is ohmic heating or moderate electric fields impact protein structure, interactions, and aggregation. The electrical field strength and frequency using this during these processings are critical processing parameters. The effects that we observed at molecular level result in a cascade of events resulted in differentiated functional properties. And of course, moderate electric field processing has the potential to tune and control biotechnologic processes involving proteins, their structural stability, interactions, supramolecular structural formation, and among others. And that was it. I would like to finish by thank you all for your attention. And of course, my team, Professor António Vicente, Ricardo Pereira, José Teixeira, and our research group at Food Innovation and Technology, and also Industry and Bioprocess Laboratory. Thank you so much.
Thank you very much, Rui. That was fantastic talk, actually. Very timely to what's happening also in our team. So a lot of interesting data. So therefore, I believe there'll be plenty of questions. So let me see if, at least I have, definitely, I guess somebody from
Great.
attendees will as well. So if there is any raised, you can raise your hand or well, the attendees, or just type into the chat if for any reason you cannot. Okay, so I'll start meanwhile while other people think. So I think my major question is, maybe I'll just double check first. So I understand this treatment, how long typically does it take? You know, it's a scale of the seconds or minutes.
Well, yes, that actually can be adjusted according with the intended purpose. For example, if you are using moderate electric fields as food processing strategy, we follow the standard thermal temperature binomials as a pasteurization, ultra-pasteurization, and so on. So we can, for example, reach 90 Celsius for 1 second, and that's the standard for high temperature pasteurization.
Okay, 90 seconds, okay.
If we are, for example, promoting protein aggregation, we know that the aggregation process takes some time. We can prolong this treatment for a few minutes, up to 5, 10, 15 minutes, in order to reach the aggregation level that we want to.
Okay. The reason why I'm asking, and this is really fundamental for the mechanism, is that if you are losing, and if you also demonstrated that the lower frequencies tend to have more effect by the reasoning you have, my biggest, well, say, interest would be to what extent do you control the electrochemical processes? Because if you are low frequencies, you will inevitably generate, you will release metals from electrodes or whatever. Well, depends on electrodes you use. Depends on the voltages especially, but I believe that for, so what are the voltages you operate at? Are they, let's say, about, I guess they are about a few millivolts, right? Because you need to get enough steel strength. So
actually, this is actually dependent on the conductivity of the sample. When we are dealing with food stuff, well, it has an inherent conductivity depending on its composition.
Right.
If we are talking about a more fundamental study with just a protein in solution, we can actually control the conductivity and control the voltage that we apply. But generally ranges from a few volts to a few tens of volts per centimeter.
Okay, so we are always above some thresholds for electrolysis.
Yeah. Okay. But a quick note, those studies on denaturation, let me just come back, that we probe the denaturation profile process in situ. Here we actually use a really low voltage, and some of these voltages were actually below the threshold for the electrochemical reactions. Another thing, the use of high frequencies is highly effective to mitigate this issue, because when frequencies go above, and this is documented, when frequencies go above 10, 15 kilohertz, the polarity changes is so fast that the polarization on the layer next to the electrodes does not take place. So the electrochemical reactions are effectively eliminated. So yes, using low frequencies, we have to deal with electrochemical effects. At higher frequencies, this is no longer an issue.
Yeah.
And in fact, a few years ago, I did not include it on this presentation, but we conducted a study probing exactly that. We use different electrode materials, such as stainless steel, titanium, or graphite, and we use different electrical field strengths and different frequencies, exactly to probe electrochemical reactions, metal release, and how that will affect, for example, protein structure and protein aggregation. And there we are able to actually prove that more stable materials are less problematic because, for example, titanium is the amount of metal released to the solvent is really low. And of course, using graphite, no metal was released, but we still could observe some of these effects in protein structure.
Yeah, well, that's very. I'll follow up. Great, thanks a lot. I'll give space to Sandra Costanzo. So Sandra, I'll make you a presenter so you can unmute and ask yourself if you prefer. So Sandra, if you like, go ahead.
Okay. Morning. Hi, Michael.
Hello.
Hi.
Hello. Good to see you.
Congratulations. Very interesting talk.
Thank you. Thank you so much.
Some curiosity. I liked that. I liked correctly the difference. The difference in is regarding the effect on protein when applying low frequency and high frequency electric field. And in particular, you also say that if you have a specific condition, when applying higher frequency, the proteins are not so forced to align with the applied electric field. But is there a condition we can achieve from a theoretical point of view in order to have the same alignment is at low frequency? Maybe it can depend also on the amplitude of the applied electric field.
Yes.
So I ask just this.
Yes. I'll try to move to that part of the presentation. So yes, in this particular case, and the studies conducted on this matter were actually just probing the frequency effect. And the electrical fields applied were actually very low to minimize just the polarization effect. But yes, in theory, if you increase the field intensity, this polarization will be more be faster, and in theory it should be a possibility to also control this molecular motion through the field intensity, of course.
As the possibility to apply an orientation for the proteins may be useful in some biomedical applications, such as
yes, actually,
cancer.
it's something that we are very, very interested in, and we are just starting to explore that. But of course, we believe that there's a lot of potential there. If you can control orientation, the implications are huge, of course.
Very much implication. Thank you again.
Thank you for your question.
Hi, Michael.
Okay, so good. So yeah, and one thing beyond, if I may follow up what I ask, the metal release is of course one effects of electrochemistry, and the hydrolysis of course depends on the current as well. For example, we also in our recent paper we used glassy carbon electrodes where there was of course no metal to release. However, they turn out to be very good producers of reactive oxygen species, and these are harder to probe. So you can only see, you can see indirectly, you have more oxidation, which can be seen as less SH groups because they are, well, in general, the reactive oxygen species can promote, let's say, disulfide bond forming, but it's much more complex, of course. So I'm wondering, have you ever looked on these oxidative aspects of caused by electric field exposure?
Yeah, great question. And the answer, yes, we try to look into that. And of course, it's something that we should consider. And as you mentioned, they are quite hard to probe. A few years ago, we made an attempt with the infrared spectroscopy to detect these oxygen species, but without much success, because they form and they react quite quickly, and
exactly.
Mostly we can deal with the consequences, not with their formation directly. And yes, we conducted some studies changing the conditions, either again voltage, frequency, and so on, in conditions that we know that we were promoting electrochemical reactions and the formation of reactive species, and the other conditions where we knew that this was not occurring. And we were actually able to see some small differences in not only protein structural features, but as you mentioned, in disulfide bridges and cysteine availability. Actually, we did that using fluorescence spectroscopy because tryptophans are great molecular probes, and the main protein of our study is beta-lactoglobulin, and we know that there's a tryptophan inside that beta barrel next to a disulfide bridge, and by connecting the different behavior in this tryptophan positioning, we could infer its interaction with the neighboring disulfide bridge or free cysteine, and we are able to observe some correlation actually. So which suggested that low electrical frequencies and the reactive species form may actually help to break that bond.
Okay, I see.
But not very conclusive. It was suggested that way, but we could not probe it in detail.
So maybe in the detecting ROS, we can help out because, well, we have a technique which we worked on since a long time. It's these endogenous chemiluminescence, which basically you can do real-time measurements of the ROS production as you pulse or as you apply electric field. So that's something
great.
Which could be interesting. And one of the main authors of the study is also raising up the hand. So Michal, I'll make you presenter. Maybe you have other question as well. So Michal, go ahead. You can unmute yours.
Thank you. Yes, thank you very much for a nice presentation. Please, all the results you have presented, the frequency resolution of your plots and graphs was not very high. So I wonder if you were considering also some resonance effects in terms of frequency.
Well, actually, we did not. We know that resonance frequencies for proteins, it's actually in higher frequency levels that we use in our studies. That's actually quite interesting, but we don't use these frequency ranges. The main reason is you use electrical field processing well as a processing tool in food and biotechnology, and the commercially available equipments, or the ones we can construct, operate under these frequency regimes from 50 hertz to 25 kilohertz usually. So in practical applications, these are the frequencies used in this kind of technology. And because of this reason, we have focused in this frequency regime.
Okay, thanks.
I actually had a similar question in mind, not necessarily with the resonances, but yeah, the time scales are selected. Understand the technical reasons, of course. The higher the frequency you go, higher the voltages becomes more complex and more expensive. That's clear that for applications when I have something accessible to companies and to users. So anyway, now just from the mechanistical perspective, where we try developing tools to understand what's happening. So when we're speaking about the time scales of the protein rotation, this frequency which matches the amplitude of motion, I guess it's the period of that wave would be corresponding to something like well, or maybe this is my question: is it corresponding to rotational diffusion time scale of a protein, which will be dependent on its basically volume and the viscosity and temperature? Is it that
exactly
what you had in mind?
It depends on the molecular size of the protein, its overall charge, of course, and viscosity of the medium. That's absolutely right.
Yeah, that's
so this will be this will be protein specific and also specific to the conditions that we are using.
Okay, because that will be for, well, say, let's consider something like medium-sized 60 kilodalton. Well, that'll be corresponding to, I guess, to some hundreds of kilohertz, roughly. I guess maybe for small protein in higher frequencies. So yeah, just thinking like if you could decide that really have, well, any generator, or we would like to have, what would be the optimal frequency to use, or are there other parameters to consider? Of course, interaction with the substrate, if it's enzyme, that matters a lot. And other stuff. So it's quite complex, but I'm trying, this time scale of the motions was I'm trying to understand if the same perspective we have here.
Actually, this study is not it's not from our group. It's from the group of Professor Sastry, which we sometimes collaborate with. As I recall, their publications on the subject, they actually mentioned low frequencies on the range of 60 hertz or 100 hertz. So these are really low frequencies. And as I recall, they relate the amplitude of motion with the intermolecular interactions of water, maybe. But I have to double check that.
Okay, because I'm always trying to connect, of course, the structure to the phenomenon. So also its interactions with the surroundings. So, but something, of course, now this might be interesting to see in molecular dynamics simulations. However, in all the MD simulations, because it's very hard to capture time scales longer than microseconds. So this is, I think, why it's kind of a gap between the understanding of these lower frequency effects and well, time scales, let's say microsecond, nanosecond, because it's simply hard to capture on a molecular scale.
Exactly.
One thing I also was very interesting for me when you mentioned the energies. There's also probably from a Sastry group. It was one of the slides you have, the kilojoules per mole or something like that. Yeah, exactly. It's the same slide. So let me understand. So if there is a this is the, let's say the force is applied at 5 volts per second, but this is the energy which is distributed over 1 second, right?
Yes.
So this, I think, matters a lot, because what you really need to usually, well, classically, the imagination breaking the bond is probably you have to have energy focused on one spot at particular, let's say, very short time to break it.
Very short time, yes. And this is a cumulative effect reported here.
Yes. So that's, of course, by applying the external forces, you're shifting the equilibrium of all the well, interactions. But still, I mean, is it interesting to mention? But also what is the quantum of the energy, which is here what I think might be determining. But I really appreciate it. You also open us for us, let's say, maybe let's say new perspective, because I know there is a lot of interesting literature in food industry. I'm just not familiar with the, let's say, with the groups and with the people doing that. So it was also interesting to learn about where to maybe to look on in better understanding what's happening. But yeah, that was really, really interesting. Okay, yeah. So very, very, very interesting. So I wondering what else I would, because there are many technical details which you mentioned here. We have similar things which we've been using. We've been also using circular dichroism, fluorescence spectroscopy, ANS, Elman reagents. So very similar techniques which we also learned from the papers from the literature and implemented into our stuff. So yeah, I mean, typically what we were really trying to interesting to dissect was, because we were mostly working in rather isothermal levels. So
exactly.
Our goal was to not to change the temperature, just to, because we are coming from, let's say, bioelectromagnetics community where everybody's like freaking out, of course, if you raise the temperature by more than something. So what is it affect? So I understand, however, these things can be compound and synergetic. So we cannot just we should not discard it. And also it's important because it has history on of the food processing, right? Thermally was something standard thing to do. But I love that you show that it depends how you heat.
Of course.
It matters. So, but however, our specialty in our team is really on looking on dissecting the contributions which are due to the electrochemical effects, which are there as long as you have the Faraday current flowing,
of course,
and how to monitor that well efficiently and fastly. So that's why I was really curious about these in these effects. So if, for example, because maybe it's possible to distinguish it from the nature of the data. For example, for if you're using Elman's reagent, have you ever seen opposite direction in terms of if you have increased SH group accessibility or content, so to say, or decreased?
Yes.
Let's say, because that's something where this is going off in another direction. So if you see the data, yeah, go ahead if you have any comments on that.
Yeah, well, of course, we look at this as a processing perspective, and the thermal increase is part of the food processing and some bioprocessing methods, of course. And for us, it's a challenge to differentiate these effects, thermal, electrical, electrochemical, for sure. And that's been on the core of our concerns for a long time. In this kind of study, we always use, for example, try to keep the temperature kinetics as close as possible. So using the same heating profiles, and sometimes change those, because with electric fields it's quite easy to change the heating profiles. With a conventional heat exchange, because it's limited by diffusional problems, that's not so easy. And we actively consider that, of course, try to differentiate what is electrical effects, what is electrochemical effects, and what is thermal effects. But in the end, it's a synergy between all of them, of course.
Sure.
And regarding using the Elman reagents, yes, we have seen interesting patterns under specific conditions, where we increase or decrease the cysteine availability, of course.
Okay, I see. Yeah, I mean it's for thermal, let's say, effects, and the and let's say physical effects are kind of, I mean, it's maybe way of will be in the end some product for a customer, or in the end the consumer. I think they would like to know if the things are, if they are, it's just, trade word, chemically changed. So if you have a processing which is purely physical, so purely thermal, or electrical, then it will be only changing basically the conformation. It will open up, closing, and aggregating, and many other things. However, if there is electrochemistry, there will be chemical changes, which of course can be same level of effect, and people might be maybe more scared of that, but maybe not, because it's anyway you're doing, if you increase temperatures or increase any auto-oxidation which is taking place anyway. So that's another thing to consider. But I think these I'm always been considering myself like for a food processing, like okay, so maybe put differently, will be I'm very curious about is to try to say, okay, so in this type of conditions it is 30% electrochemistry cost, 20% heat, and it's probably synergetic. You can really decompose to the individual components, but at least differentiating between the physical and chemical effects of the electric field treatment. That's something they're trying to dissect, and it's of course very complex because it's It's energetic.
Yes, yeah, that's actually great observations. And just to clarify some aspects, at research level, we keep, for example, frequency as a variable because it's very interesting to try to understand how things going.
Mm-hmm.
Commercially, in terms of food processing, the first commercially available equipments were operating under low frequency, 50, 60 hertz, because it's the frequency
Right.
of our power supply.
Right.
But recently, all manufacturers are using high frequency, usually 20, 25 kilohertz. And
Okay.
on that sense, the electrochemical issue is resolved or minimized because in these frequency regimes, the electrochemical phenomena are almost neglectable.
That
And this,
depends
of course,
on the current. I mean, I think
yeah.
this is the mantra, which I also knew, but go ahead. I will follow up on that. Go ahead.
Okay, thank you. And of course, there's significant amount of studies regarding chemical modifications and safety of food processed by these kinds of technologies. And of course, this is concern number one. And so far, pretty much all the studies that I'm aware of ensure that safety and not extensive modification. But just out of curiosity, on those studies that we conducted on immunoreactivity of allergenic proteins, I didn't go into much detail because the combination of temperature, electrical fields, and frequency, and metal release from the electrodes, it's actually quite complex. But we found that in some conditions where we promoted electrochemical reactions and we expected some chemical modifications of the proteins, were actually very effective in reducing protein immune response.
Okay, interesting. I mean, it's all tools, right? In the end, so you can use it as a tool if it works the way. Oh, yeah, that is very interesting. You definitely will follow up. I don't want to hold you for too longer, but it's I maybe one more comment for this. So yeah, this frequency dependence of electrochemical reaction, that's indeed true. However, that in the end will depend on a total amount of the Faraday charge transfer. That means even for higher frequencies, if the currents are high enough, there will be some electrochemical product, and it really depends, as you mentioned, the conductivity. Well, that determines the current in the end, but also the materials of the electrodes, and in the end, material of the food material there. So yeah, but I think what's very common and could be controlled for as long as you know, if you will see an oscilloscope, if you have an oscillating field, you could see the phase shift between the phase and current. That will tell you if what's the nature of the current, if it's capacitive or Faradaic. If it's in phase, you know, purely for Faradaic, purely resistive, it means all the current goes there is going to the Faradaic current, more or less. So, and that has to generate something, chemical changes. So this is something which it can be rather easily checked, like even with the effects are not seen, might know if, okay, am I generating electric current, Faraday current? Of course, a lot of the things can just react fastly on the electrodes and will not touch the, I mean, the electrochemistry can just back react or, you know, recombine products, or electrochemical products can recombine on the electrodes, depends really on the materials, and they will not chemically affect the material in the cell or in the reactor. So, but yeah, this definitely, there are interesting stuff we should chat about, like follow up.
Yes, of course, definitely, yes.
Okay, let me give you any more space to the attendees before I stop the recording. Anybody else like to ask before we go record? So if it is not for the recording, I would like to thank you, Rui, for a very interesting talk.
And thank you for the invitation.
I'll stop the recording now. Thanks to everyone as well.